everyone, and welcome to today's live broadcast. Live single-cell phenotypic biomarkers measured from primary biopsy samples, an emerging class of biomarkers towards the development of precision diagnostics and discovery and development. Presented by Ashok Chander. He is the co-founder and CEO of Cellanyx. I am Marjorie Torres of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the green Q&A button located in the lower left of the presentation window and type your questions into the box that appears on the screen. We'll answer as many of your questions as we have time for at the end of the presentation. Also, please notice that you'll be viewing the presentation in the slide window. To enlarge the window, just click on the screen icon located on the lower right. If you have trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window, or use the Q&A button to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the button in the bottom left corner and follow the process to obtain your credits. Please join me in welcoming Dr. Ashok Chander. I will now turn the presentation over to him.
Thank you, Marjorie. This is a great pleasure to be able to speak to you around this topic of live cell phenotypic biomarkers, specifically from primary biopsy cells. And what I'll present to you is evidence to suggest that these are a new class of biomarkers that are useful for precision diagnostics and the discovery and development of personalized therapies. Specifically, what I'll go through is why machine vision and machine learning are extremely important for this process towards understanding live cell dynamics as well as tissue, co-culture, and single-cell resolution. So we all are very well aware of the need for and the importance of precision diagnostics and personalized therapies in solid tumors. We've been subject to classic techniques that have yielded the results that we see today. And a lot of these types of cancers, lung, prostate, breast cancers, for example, are still very much in need for personalized therapies. For example, in prostate cancer, castration is still a viable mode of treatment. So in order to really get at personalized and precision medicine, we need to think about approaching current diagnostic and current drug development techniques in a different way. Specifically, the problem, as we're learning, is, of course, the idea of tumor heterogeneity. Of course, not only is there differences between the cancer within patients, but there's also differences within the same patient, as well as, for example, in a prostate, two different lesions within that prostate will have different molecular profiles, and then even within one profile, there will be variations among cells. So what we're finding is, and what everyone is certainly excited about, is the prospect of being able to analyze and interrogate biopsy samples, tumor tissue, patient samples on a single cell level, but not just from one level of regulation. As we know, cancer is a very complex problem. And so perhaps we might want to apply the full central dogma to it, or interrogate the full central dogma to it, and not just one level or aspect of it. Towards that aim, Cellanyx, the company that I represent as CEO and founder, developed a platform, what we term STRAT-X. And this platform enables the culturing of primary biopsy cells for automated multi-omic, and what I mean by that is phenotypic and genotypic, proteomic, level type, high throughput and high content analysis. So let me walk you through what the STRAT-X platform does. Quickly, the STRAT-X platform is able to take a biopsy cell from the OR. That biopsy cell is able to be transported, given a media formulation, from the OR to a central laboratory within the hospital or external. And then that biopsy sample is dissociated into various levels of resolution to be able to be placed on a microfluidic device that then can be imaged through through microscopy. Once the biomarkers and images of interest are collected, machine vision software can then simply analyze objectively and quantify biomarkers from those digitized images, and seamlessly those quantifications then get input into machine learning derived statistical algorithms that can generate predictive metrics that can facilitate the understanding of clinical outcomes such as metastasis, local progression, therapy response, and even upstream of that, biomarker validation. And so the point being, there's versatility both in the front end of the platform as well as in the back end, in as much as number of biopsies can be taken and cultured, and then a number of predictions can be had. Importantly, this process is actually quite quick and automated. So the most labor-intensive step is, quite frankly, taking the biopsy out of the box that it's been received in, and then placed into a buffer that then moves the process along, moves the sample along this process of an incubation and then a normalization process. It's normalized specifically to an extracellular matrix formulation that not only keeps the cells alive, but then promotes the cells to react in very specific ways that might be interesting for the prediction of interest. Then there's the sample imaging, which can be anywhere around 5 hours, and then the data analysis, which is done on the cloud, that will yield predictive metrics. Here's an example on slide 7 of the way that cells look within the platform or in the microscope. We have primary, actually an example is here's a primary biopsies of prostate cancer cells. Each cell is actually identified and tracked by the machine vision software. And then during that tracking, of course, biomarker measurements can be measured through the machine vision algorithms. If we can play this video, that would give you a better sense of how dynamic these cells are, and also one might be able to appreciate how many biomarkers and how many dynamic biomarkers there are that add a tremendous amount of value to the analysis of these cells. So perhaps we'll get back to that video, but just okay. So to go back to the that brief clip of the video, perhaps we can play that a little later at the end of the presentation. But the idea of that video is, and the analysis, of course, is to measure dynamic biomarkers and then associate them with molecular and perhaps more traditionally static biomarkers. And so on this slide, slide 8, we see that a kind of a representative grouping of some of the biomarkers that can be measured on the STRAT-X platform. And so, for example, we see adhesion. Again, these cells are interacting with a very defined extracellular matrix, so their adhesion characteristics can be very telling with respect to their integrin and the ways that they might generally interact with the extracellular matrix. As the cells spread in the in vitro environment, we can also assess the morphology and the molecular dynamics as well as the cellular dynamics that also give really good insights into specific ensembles of protein complexes such as the RAC, Rho, and GTBase complexes that govern actin signaling. After the live cell dynamics are measured, the cells are fixed, and then the cells are probed for fixed and proteins to look at, or even RNA to look at localization, expression, and modification states. And what's interesting, though, is that when, after all of these analyses are done, we're able to assess these biomarkers and create aggregate biomarkers, proprietary aggregate biomarkers that actually are pretty interesting and useful in terms of clinical predictions. And I'll get back to that a little later. So just kind of thinking about it from a high level, the STRAT-X platform is very adept at measuring both dynamic and fixed biomarkers. And one can think of these as multi-omic biomarkers, cellular and molecular, specifically dynamic cellular biomarkers and fixed cellular biomarkers and dynamic molecular and fixed molecular biomarkers. And it transverses the levels of regulation that are responsible for the disease and pathology that patients experience, from DNA to RNA to protein to cellular interaction with the extracellular matrix or with other cells. So the power of STRAT-X is also based on the ability to measure multiple different types of samples. And so what we're talking about is not just the ability to measure from biopsy cells, but we can measure from blood and urine. Cells derived from these samples have interesting touch points within the treatment paradigm. Specifically, at least traditionally, blood and urine are adept and great candidates. Those samples are great candidates for screening technologies or screening applications. Conversely, biopsy samples are more adept at maybe diagnostic or prognostic applications. And so the point here is that simply live cell phenotypic diagnostics and live cell phenotypic biomarkers can be toggled or applied to various applications within the treatment paradigm, largely, I think, based on the sample input. And the value of these biomarkers is what we'll get into a little later. But certainly the increased sensitivity and specificity of our predictions within the diagnostics and prognostics touchpoints could easily be replicated and recapitulated in the screening aspects as well. So here's just a quick overview again of the STRAT-X platform, and then tying it to specific predictive metrics. So well, moving forward, we'll be talking mostly about the prostate, and a prostate biopsy is taken. Presumably, it's taken from the lesion. That core has been placed in a tube filled with culture media as opposed to formalin-fixative. That tube is then packaged on ice or cold packs and transported within 72 hours to a central laboratory where it can then be processed by dissociation, normalization, and then loaded onto a microfluidic device for image analysis towards biomarker quantification by machine vision. And then, of course, after those biomarkers are quantified, they're put into machine learning derived statistical algorithms to predict. And so the example that we'll be moving forward with is the ability to predict whether a patient experiences surgical adverse pathologies. And these predictions are very useful in terms of patient stratification. For example, a Gleason 6 or a Gleason 7 patient may need to understand whether they're at risk, being a traditionally or classically defined low risk or intermediate risk patient, whether they're at risk for being or having a surgical adverse pathology, to understand whether or not to go through active surveillance or to undergo definitive treatment. And so to do that, we've created predictive metrics, and again, what we'll do is we'll get into that a bit later. Beyond just that ability to use the test as a patient risk stratification test, there are upstream applications of this platform that can also be useful for the development of precision or personalized medicine, specifically when therapeutics are being developed, the ability to rapidly culture primary cells as opposed to using cell lines, or as opposed to using animal models, can be very important. The ability to look at multiple levels of regulation, or that multi-omic approach, could also be very important towards understanding the complexity of the diseases that we're trying to target, as well as the high throughput analysis of machine vision and the predictive nature of machine learning. And so in addition to being a risk stratification test, this STRAT-X platform has many upstream applications with respect to therapy selection, and actually some downstream applications with respect to patient treatment in terms of monitoring if a patient is actually responding to a therapy or a treatment, and actually deciding whether that treatment is appropriate before the patient undergoes that treatment. So here we are in terms of looking at the first major key capability of this platform. And this platform is hinged upon the ability to keep primary biopsy cells alive. And in the panel on your left, you'll see various groupings of controls: glass, polylysine, and laminin, classic surfaces that people culture cells on. When we coat these, when we coat glass or plastic with the STRAT-X ECMF, or extracellular matrix formulation, we see a very robust response by primary biopsy cells to adhere and survive and behave in a meaningfully referenceable way. And we've shown that this ECMF, or extracellular matrix formulation, can not only keep cells alive across multiple days, but even beyond over the course of 30 days. We haven't tested beyond 30 days, but it's exciting to know that we can rapidly culture these cells, and then they can survive in a relatively long period of time. So at least in the context, in the application of clinical diagnostics and therapeutic development. So now that we have the cells alive, we certainly want to make sure that we can objectively measure them and quantify important biomarkers or features or behaviors of these cells. And I think it's important to note that the objective nature of this quantification is kind of critical to the next steps of the platform. Without consistent objective measurement of biomarkers of interest, the machine learning algorithm will just not behave or be trained properly. And so the old adage goes, garbage in and garbage out. So we want to ensure that the data that is being input or that is actually being observed and measured from the STRAT-X platform is as objective as possible and as robust as possible, such that the machine learning algorithms can be as smart as possible. So once we are able to actually employ machine learning, then a whole new world, if you will, opens up. We can take live cells, we can choose which biomarkers are of interest to us, given a vast suite of again, multi-omic biomarkers. And then rather than us or the operator choosing those biomarkers of importance, we let and train the machine learning algorithm to decide and arrive at an appropriate ranking of those biomarkers. For example, if one is going to predict metastasis in breast cancer versus metastasis in prostate cancer, given the same set of biomarkers, again, a very vast set of biomarkers, it's the hypothesis, of course, and what we've proved to be is, and we've shown and demonstrated, is that those biomarkers are very different. But we did not pick those biomarkers. Rather, the machine learning algorithm decided upon those biomarkers. And there's also the statistics behind that are just very overwhelming, and leads to further predictions and further intelligent predictions, both by the operator and by the machine. These predictions can be very vast and varied, not just from a prognostic approach, but again, upstream in terms of therapeutic development, as well as downstream in patient care. So on this slide, we see actually the value of this approach. And so as an example application of STRAT-X, we've demonstrated that this platform is able to predict surgical adverse pathologies for prostate cancer. And again, that's the gold standard of prostate cancer diagnosis, in as much as the prostate is removed and interrogated by the pathologist to understand what exactly happened within the prostate. And being able to predict that, certainly before the surgery, is a very important, and one might consider the holy grail of prostate cancer diagnosis. And so what we've done is we've taken 238 samples, patient samples, and predicted that with relatively high sensitivities and specificities. And this data was presented in the American Urological Association in 2016. And received a Best Poster Award for a prostate cancer biomarkers for risk stratification. What's also interesting, and kind of going back to the point that I may have alluded to earlier, is that given this breadth of biomarkers, one can make multiple predictions with different rankings of these biomarkers. We were also able to predict, using STRAT-X, surgical adverse pathology features, even if the tumor, or rather the biopsy, missed the tumor. And so, of course, in the field, or in the state-of-the-art rather, this is termed the field effect, or around the tumor there is a region of tissue that, while might not look like tumor from a histopathology standpoint, there are molecular changes that occur in that tissue that define it as abnormal, essentially. And this suite of biomarkers that STRAT-X actually was able to pick out those patients from the field effect that would actually experience adverse pathology. And so, again, just using this as an example and why we should think about live cell phenotypic biomarkers and live cell multi-omic approaches as being meaningful, we can compare this approach, this STRAT-X approach, with other fixed cell and genomic approaches. And so, as a previous slide alluded to, in terms of prognostics, the Gleason score is roughly 60–70% accurate as a prognostic. In terms of prognostics and being able to predict surgical adverse pathologies, genomic approaches also, just purely based on their biomarkers, are sub 80% accurate, and so just above clinical noise. We find that in our results, with similar patient numbers in terms of our clinical trial, these dynamic biomarkers are actually achieving better performance with respect to prediction. Not only that, but it avoids repeat biopsies by being able to predict even if the tumor, or even if the biopsy misses the tumor. It's faster and efficient in terms of workflow, which I'll get into a bit later, and it turns out to be a little more cost effective. So in terms of workflow, we see that the Gleason score usually takes around 7 days for a pathologist to return that. There's a number of processes involved in terms of preparing the biopsy core and then staining it and then observing it. In the genomic workflow, it takes a little longer, specifically because the pathologist is asked to identify tumor-rich cores to send to the genetic analysis, to send for genetic analysis. The CELLANYX workflow actually is simplified, and one might think that it's more complex given that it's using live cells. It's actually simplified in as much as, because the STRAT-X platform only needs one biopsy, in as much as it can predict from tumor or adjacent or normal tissue. It just needs one biopsy core. That biopsy core can then be placed in a media that then is put into a transport box filled with ice packs, and then sent to a central laboratory. And that workflow has resonated quite nicely with the practicing physicians that we've shared that with. And so what we're seeing now is, and what's emerging, and what we will see, is that 100 years of using histopathology, and the some odd 30–40 years now that we're been really excited about the promise of genomics, may yield to an actual multi-omic approach. And while the discovery of DNA in 1960 was very exciting, and while that afforded us a tremendous amount of advances in information in biology, it wasn't meant to actually prevent us from taking the next step in the central dogma and understanding the other aspects and other levels of regulation towards really precision, or to really developing precision diagnostics toward personalized therapies. And so what we see here is that the STRAT-X platform, as an example of a live primary cell platform, has many applications. We can use it in biomarker validation, therapy selection, patient stratification, and then certainly those being part of the therapy, the drug development pathway. And then in the clinical landscape, we can think about it as being a diagnostic, a prognostic, or a companion diagnostic. And so here's some data just to kind of give you an example of how all of this is working and can work. So key to the STRAT-X platform is the ability to actually interrogate biopsy samples with single cell resolution. So on the left you see a graph of each point being a single cell and their measurement for one given biopsy. And these cells that are above the dotted line are predicted as being positive by the machine learning algorithm. And one can understand how if one would add a drug and then push all those cells down below the threshold, one might have a very positive and predictive and useful drug on their hand. So similarly, we can place this general concept of single cell resolution and translating that information into a patient level prediction into other types of useful analyses, specifically within drug development. For example, biomarker identification or biomarker validation. One, of course, would know the clinical endpoint that the biomarker is going to be useful for, and so one can start probing different biomarkers and understanding those perturbations and whether they actually affect the prediction of the clinical outcome that is needed. And again, this is a two-step process in as much as one would predict patient behavior based on single cell results, and then by perturbing those single cell results, see how that shift in patient behavior occurs to understand whether that specific biomarker or that specific compound is of interest. So kind of a very simple way of thinking about it is the STRAT-X platform is able to predict whether a patient is positive or negative on the x-axis for a given adverse pathology or a given outcome. Let's call it metastasis. And so if this patient is negative or positive, it determines whether the patient needs therapy or not. On the y-axis, we see that this platform, STRAT-X, can also predict if the patient, when treated, would be positive or negative. And so essentially the platform can be applied to predict the responders of therapy by identifying those patients that would be positive for metastasis, and then also be responders of the therapy in as much as that metastatic behavior of the cells would be diminished, if you will, by the addition of the therapy. And that's, of course, in the bottom right quadrant of that graph. So that's just a quick look into some of the capabilities and applications, I think, of live cell phenomics and live primary cell phenomics, and the ability, of course, to keep biopsy cells alive, measure biomarkers objectively with machine vision, and leverage machine learning for important predictive metrics, certainly has a tremendous amount of application across both the R&D as well as clinical treatment paradigms, not only within oncology, but in other disease states, specifically fibrotic diseases, as well as even cardiology and metabolic diseases. So in summary, what I just wanted to kind of emphasize is that it's the rapid culturing of these cells that makes things meaningful, both within the context of developing drugs as well as in the context of clinical diagnostics. Then certainly it's the versatility of being able to co-culture not just single cells, but different types of cells together, which aids in the ability to understand cell-to-cell interactions for those similar applications. High throughput analysis, and then most importantly, the objective analysis by machine vision and the predictive capabilities of machine learning really take and move what might be considered a very old idea of phenomics into a very relevant and actually perhaps even more important stance. And beyond just kind of the tools aspect, we see a tremendous amount of potential with therapeutic development throughout the pipeline of biomarker discovery, lead compound development, stratifying patients within clinical trials, understanding patient response, and then certainly in the context of clinical diagnostics, as the data that we just talked about supports. So I do want to take a quick moment to recognize the team that built STRAT-X'S. Michael Manak is the primary cell biologist, and Jonathan Arsenik is the primary computer scientist. We've had a great number of important and thoughtful advisors, starting on the bottom right, Sam Cia, professor at Columbia University, involved in microfluidic design and diagnostic development. The chair of urology, Eric Klein, of Cleveland Clinic, Dr. Eric Klein. Kevin Knopf, health economics and breast cancer expert. Dr. David Albala, again, a chief of urology at Krause Hospital in New York, and the former chair of urology at Tufts, Dr. Ghanem Sant, and Dr. Pravin Chaturvedi, who's an expert at drug development and lead candidate optimization. With that, I'm happy to take any questions. Thank you.
Thank you, Dr. Chander, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the green Q&A button at the lower left of the presentation window. Type your questions into the box that appears on your screen and click on the send button. We'll answer as many of your questions as we have time for. Let's get started. Our first question is, what are the key technology features that enable live cell phenotypic analysis?
Yes, the key features, I think, are one, of course, the ability to rapidly keep cells alive, and not just on the device or not just on top of the microscope, but in transport. And then, of course, the ability to have a good suite of biomarkers that are of interest to whatever general area or indication one is interested in. And then 3, of course, the machine vision that we talked about towards objectively quantifying the biomarkers. And then finally, because there may be a number of biomarkers and a number of systems being interrogated, the machine learning helps a great deal towards prioritizing what actually is important towards the prediction of interest. And so it's a hard question because they all have almost similar weights. But I would say that of course one won't work without the other.
Thank you for that answer. Our next question is, what are the most promising applications of utilizing live cell phenotypic biomarkers?
Yeah, so I'm not sure if I heard that question properly. Can you repeat that question? Sorry. Okay, so
Our next question is, what are the most promising applications of utilizing live cell phenotypic biomarkers?
Thank you. So I think the most promising applications of phenotypic biomarkers are the ability to risk stratify patients, because currently there are no precision tools to do that. This is the biggest unmet need, I believe, in clinical treatment as well as even drug development. The ability to understand which patient has a severe defect in a pathway, or has a or even as general as is going to have a metastatic disease or a locally aggressive disease. I think this is the biggest need for this type of test, in as much as the ability to do that will then allow for other advances in other areas. For example, the ability to properly risk stratify patients will make it easier to develop therapies, and the ability to properly risk stratify patients will also enable better treatment of patients. So I think the most important and promising application of live cell phenotypic biomarkers, in as much as diseases are dynamic, diseases are complex, the understanding of dynamic biomarkers may add important information to that understanding, is really an actual risk stratification and patient kind of categorization. Thank you.
Our next question is, how long does it take to analyze a sample?
Thank you. So I think generally it depends on how many biomarkers one wants to measure, but in our experience, it's been 12 hours to receipt of sample. We pushed that out up to 72 hours just to test our ability to do that. And then beyond actual sample receipt, the disassociation and normalization process takes another 12 hours. roughly, and then the analysis can take anywhere between 5 to 10 hours, depending on what one might want to measure. And then the data analysis can be condensed to 1 hour, depending on computing power. So we're looking at within 2 to 3 days, whereas the current assay systems are potentially over weeks. So this is a big advance in and of itself, on top of the actual advance of being able to measure dynamic biomarkers.
Okay, it looks like we have time for one more question. Our last question is, what contributes to the predictive power of live cell phenotypic biomarker analysis?
Thank you. That's a very important question, and one that I don't really exactly have the answer to at the moment. Again, I think I'd have to go back to just the idea of looking at multiple levels of regulation of the cell. The ability to not just look at one aspect, or DNA or RNA or protein or even cell, but to look at multiple levels of regulation, but then also look at them in a dynamic way, given the fact that these diseases operate in a very dynamic fashion. And then I think that flexibility is leveraged by machine learning. And so then I think my lack of understanding of this perhaps is because to understand all of the biomarkers that are being assessed by all of the machine learning is very challenging, perhaps even for my simple brain. And so the ability to leverage artificial intelligence, as it were, to then sequentially measure and analyze and compare thousands of biomarkers over thousands of cells, essentially 45 million measurements per sample, and then come up with a ranking of those biomarkers over the multiple levels of regulation again. I think that is that systems approach and that computational approach are the two keys of its predictive performance. Thank you.
I would like to once again thank Dr. Ashok Chander for his presentation. I would also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through January of 2018. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us, and we hope to see you again soon. Goodbye.